so honored to be speaking amongst other scientists for whom I have such great respect. The work that I will be talking about today was part of my PhD thesis work, which I completed at Woods Hole Oceanographic Institution, where I was co-advised by Lauren Molyneux and Dan McCorkle. And before I start, I would like to acknowledge my funding sources for this work. So I was supported by a National Defense Science and Engineering Graduate Fellowship from the Department of Defense. We also had funding from Woods Hole Sea Grant, from MIT Hui Academic Programs Office, and from 2 internal Hui awards. So I just finished my first year teaching, and as I went through the year, I learned a lot, and 2 of those things. 1, don't make assumptions about what your audience knows. And 2, it never hurts to review key concepts. So I will be starting off with an introduction to ocean acidification, including the chemistry that drives changes in pH. Then we'll talk about factors that affect acidification, specifically in coastal waters, which is quite a different process than in the open ocean, although the chemistry behind it is the same. Then I will talk about work that I did looking at impacts of coastal acidification on bay scallop larval growth and development, and I'll conclude by talking about 2 different Northeast regional acidification efforts. So these are the results of a model showing the decadal mean sea surface pH, and it's showing that the mean sea surface pH is projected to decrease from a value of 8.2 from pre-industrial times to a mean value of 7.8 by the end of this century. And this corresponds to atmospheric CO2 levels of 280 pre-industrial and about 750 to 1,000 parts per million at the end of this century, depending on which carbon dioxide emissions scenario you look at. But what I think is most important about this figure is all of the white area. The carbonate chemistry dynamics in coastal systems are too complicated to be modeled by atmospheric CO2-based models. And so those areas are usually left out of these models. But most of the organisms that we interact with on the most regular basis are found in these coastal waters. And so it's really important for us to understand what is going on with the chemistry in those waters. But I will first start out by the driving chemistry behind open ocean acidification. So we looked at the most recent 400,000 years in the talk that we just heard, and this figure is showing atmospheric CO2 going back to the past 800,000 years. And you can see the fluctuations that we've already seen, fluctuating between around 180 parts per million to about 280 parts per million, and never going above 300 parts per million until the last century. And in fact, the rate of change most recently looks like a second y-axis, but that's actually the very fast rate of change that we've been seeing. So we can zoom in on the past 300 years and see that CO2 levels are starting to rise in the 1850s, and then in the 1950s, when most US families started to have cars and our fossil fuel emissions were increasing, the rate of change accelerates at that point. But what does this mean for the ocean chemistry? So these are the reactions describing carbonate chemistry in the ocean. These reactions are equilibrium reactions. They are always going on. The driving change at this point is the change in atmospheric carbon dioxide. So carbon dioxide is always exchanged between the atmosphere and the ocean through air-sea gas exchange. Once it's in the ocean, it reacts with water to form carbonic acid, and carbonic acid dissociates to form bicarbonate and a hydrogen ion. And the third reaction at the pH of seawater favors a reverse direction. So we have carbonate and hydrogen combining to form more bicarbonate. So the key take-home points from this is this hydrogen ion causes a decrease in pH. Remember that an increase in hydrogen ion concentration leads to a decrease in pH. But because the third reaction is going in a reverse direction, as the pH decreases, the carbonate ion concentration decreases. And that's critical for organisms that form shells, like mollusks. So calcium carbonate dissociates into calcium and carbonate ions, and this dissolution is governed by the solubility product KSP. So we can determine the calcium carbonate saturation state, which is represented by omega, and it's equal to the measured or observed products of the calcium and carbonate ions divided by KSP, which is essentially the expected calcium and carbonate ion concentrations for a given pressure, temperature, and salinity. So when omega is greater than 1, the water is supersaturated, and at that point we would expect to see, formation of shells and skeletons would be thermodynamically favorable. When omega is less than 1, the water is undersaturated, and at that point we would start to see unprotected shells dissolve. However, saturation state is also dependent on the mineral phase of calcium carbonate. So calcite is the least soluble form of calcium carbonate. Aragonite is more soluble than calcite, and then amorphous calcium carbonate and high magnesium calcite are both more soluble than aragonite. Today I'm talking about bivalve larvae, and they form their shells out of aragonite, so keep that in mind throughout the talk. So the take home from this chemistry lesson is that an increase in carbon dioxide leads to an increase in the hydrogen ion concentration. That is the same as a decrease in pH. That leads to a decrease in carbonate ion concentration, and that leads to a decrease in saturation state. And these are two different factors that can have really different physiological impacts. All organisms have a pH tolerance range that is most physiologically favorable. So as pH decreases, that has the potential to affect many different physiological aspects of marine organisms. But a decrease in saturation state is mainly going to affect the organisms that form calcium carbonate shells. And so in this case, we are very concerned about mollusks. One point that I like to make because I'm coming from Maine, Maine is very concerned about lobsters. And we don't know a lot about how ocean acidification will affect lobsters, but it's important to remember that crustacean shells are made out of chitin with some calcium carbonate to strengthen it. And so it's not necessarily appropriate to classify them in the same group of shelled organisms as mollusks for when we think about how they will respond to ocean acidification. So, what are the processes in coastal waters? Coastal waters are also affected by atmospheric CO2, but they're also further affected by coastal upwelling, seasonal primary production, and eutrophication and freshwater input. We've talked about increases in atmospheric CO2. I am going to skip over the coastal upwelling because my research is focused on the East Coast, and this is not a major factor on the East Coast. So we will talk about seasonal primary production and eutrophication, and then freshwater input. Okay, we always see an influx of nutrients from surface and groundwater discharge, but in summer months, when there's more light and the temperatures increase, we can see algal blooms which draw down carbon dioxide as they photosynthesize. But these algae eventually die, or they get eaten, and they sink to the bottom, or they're transported to the bottom in fecal pellets. And once they're down in the bottom waters, they are subjected to microbial respiration, which decomposes the organic matter. And this process uses oxygen and releases carbon dioxide. And when water is really stratified, which is often happens in the summer months when this is a major problem, we end up with spatially separated primary production in the surface waters and decomposition in the bottom waters. And this can create areas of low oxygen and high CO2 in the bottom waters. This might be something that could potentially contribute to the data that Emily showed us, showing differences in pH at the surface and then deeper into the water. So this might be a process that contributed to that. So in this case, the CO2 is not the increase in CO2 in the ocean is not directly from atmospheric CO2 increases due to human activities. It's due to the microbial respiration, but it still causes great fluctuations in the conditions that the organisms are exposed to. I illustrated this example using an algal bloom. But in a lot of coastal sediments, there is already so much organic material in the sediment that during summer months when the temperature increases, microbial respiration of that organic matter will also increase, and so we would still end up seeing seasonal cycles. So the seasonal cycles are natural, but can be amplified by human-induced eutrophication. So to illustrate an example of this, I will show you data from Wacoit Bay, which is part of the National Estuarine Research Reserve Program. And so this is Cape Cod, and we'll focus in on the Upper Cape and then zoom in again onto Wacoit Bay. So Dan McCorkle's lab at Hui has been collecting carbonate chemistry data from Wacoit Bay from 2 locations since 2008. The first location, well, they've actually been collecting from more than 2 locations, but I'm only showing you 2 today. So Menant is the more exposed site, and my symbol covers it up, but it's actually one of the 2 inlets to the bay. And it experiences some terrestrial water input, but it's also more coastally located and has more contributions from Vineyard Sound. Then Childs River is a more isolated location, which has more terrestrial influence than coastal water influence. So, okay, let me orient you to what we're looking at in these 2 graphs. The top graph shows the partial pressure of carbon dioxide, and the bottom graph shows the pH. The dashed blue lines on each plot represent the current open ocean conditions. The dashed orange lines represent the future open ocean conditions projected for the year 2100. So we can see that both locations show seasonal cycles, but the signal is much stronger at Childs River, the more isolated site. Furthermore, both locations approach the values that are currently seen in the open ocean only during winter months. And then in summer months, the more exposed site, Menant, it approaches values that are projected for the open ocean in another 85 years. And Childs River gets up above 2,500 parts per million, far, far exceeding the projections for the open ocean at the end of this century. Bivalves are found at both of these locations. There are reproductive populations at both locations. They are already experiencing these conditions, and they have found some way to handle that. And so part of what I'm looking at is trying to isolate distinct developmental stages that may be more susceptible to ocean acidification or coastal acidification. Another factor contributing to acidification or more variable carbonate chemistry in coastal waters is freshwater input. So freshwater naturally has a lower pH than seawater. And we also talked about lower pH is correlated to lower saturation state. Saturation state is also generally correlated to salinity, with lower salinity having lower saturation state. So this figure is showing you saturation state in Casco Bay, Maine, and here's Portland. And here is the Kennebec River, which has quite a large watershed. So we can see the influence of the freshwater input from the Kennebec well into Casco Bay. And this is seen by reduced saturation states. And I mentioned that when the saturation state is less than 1, that's when we would expect shells to dissolve. But this paper, Salisbury et al. 2008, they found that when saturation state is less than 1.6, bivalves start to have difficulties forming their shells. So it doesn't have to be all the way below 1 for the organisms to have difficulties. And you can see that the saturation state of less than 1.6 is found well throughout Casco Bay. So these represent current conditions. But as temperatures warm, the projections for precipitation show that precipitation is expected to increase in the Northeast in the coming century. So these conditions will only be exacerbated as we see more effects of climate change. So to review, because I'm still a little bit in teacher mode. So seasonal coastal primary production and microbial respiration creates natural annual cycles in pH and CO2 and saturation state. But human activities amplify that seasonal cycle. Freshwater naturally has a lower pH and saturation state than seawater, but increased precipitation due to warming caused by human activities will also increase freshwater input to coastal areas. So coastal organisms already cope with annual variability that's greater than the change predicted for the end of this century, and increased atmospheric CO2 concentrations will only lead to even lower pH conditions. So now I'm going to move into talking about impacts of ocean acidification on larval scallop growth and development. Okay. Scallops are epibenthic filter feeders, so they live on top of the sediment, not burrowed into it like clams do. And they span all the way from the Gulf of Mexico up to the southern shore of Cape Cod, but within that range there are two different subpopulations. On the southern shore of Cape Cod, they spawn with increasing water temperatures, usually in June and July. So let me point out this part of the life cycle of the scallops because it's pretty relevant to this. So adults live on the bottom and they broadcast spawn. The larvae, I'm gonna back up a second, okay. Ignore that for a moment. The larvae develop in the water column, so they're living in a different habitat than where the adults live. They're transported by currents and tidal actions. They have limited swimming ability, so they can mostly only swim vertically, but not against currents in a horizontal direction. Okay, as they're developing, they start to calcify between 12 and 24 hours old when they're in this stage known as the trochophore stage. When they're 2 days old, they have reached the D stage, so their shells look like a capital letter D. And at this point, the shell is fully calcified. When they're 2 weeks old, they metamorphose and they settle down onto eelgrass or another substrate. And at this point, they switch making their shell from aragonite to calcite. This could have potential implications because their vulnerability to ocean acidification could change at that point, since calcite is less soluble than aragonite. However, for the work I'm showing today, my experiments were only 1 week long, and so we didn't actually investigate this transitional period. They reach sexual maturity when they're 1 year old, and so they would reproduce the next summer after they were spawned the previous summer. Most of them will be harvested when they're about 1.5 years old, so the scallop season on Cape Cod is from November to April, so that would be the non-reproductive season. They would naturally die when they're 2 years old, but because most of them will be harvested at 1.5 years, they only have one opportunity to reproduce. Okay, so like most broadcast spawners, scallops produce millions of gametes in a season, but due to many natural events, only about 1% of those become adults. Because scallops are so short-lived and only have one opportunity to reproduce based on the harvesting schedule, the survival of every cohort of larvae is critical for the continued population of the species. Furthermore, early life stages of marine invertebrates are particularly vulnerable to environmental stressors. And we already know from other people's work that exposure to low pH and saturation state can have the potential to cause lasting effects on the organisms. And then furthermore, because the scallops as larvae are living in the water column, they're living in different conditions than what the adults experience. And so it's important to understand how they are affected by CO2 changes. So based on the vulnerability of invertebrate larvae and what had already been determined about how bivalve larvae are affected by ocean acidification, I developed two overarching questions for this work. First of all, how does exposure to high CO2 affect bay scallop larval growth and development? And second of all, is there a period in early development in which high CO2 has critical impacts on later development? So to answer that question, I designed a culturing experiment which kind of looks like a spaghetti monster here. It was completed at Huey's Environmental Systems Laboratory, and I had a very high-tech water bath to control the temperature with aquarium heaters and chillers. And these 800 milliliter culture containers are the exact same containers that you might bring potato salad home from the deli in. So it's very fancy. Every 2 to 3 days when I changed the water, I also measured the water chemistry. And so to get at the question of which particular developmental stages might have different impacts on the scallops, I designed a set of switch experiments. So initially, half of the larvae were all exposed to the same ambient conditions. And for this experiment, that was about 390 parts per million. And then half of the scallops were initially exposed to high CO2 conditions, 2,200 parts per million. If you're used to thinking about ocean acidification in the context of atmospheric CO2 change, that will look like an incredibly high value that's very unrealistic. But based on the data from Wacoit Bay, we know that scallops on Cape Cod scallop larvae are already experiencing conditions even higher than this during the summer months. So all of the experiments followed this same general trend, where at a set point in time, for half of the ambient treatments, I changed their conditions to high CO2, and for half of the high CO2 treatments, I changed their conditions to ambient. This resulted in 4 treatments, and the color scheme on the right is the same color scheme that I will use for the treatments throughout the talk. So the different experiments differed mainly in the timing of when this switch happened. So to start the experiment, I get the scallops to spawn. This can be a little bit tricky because these scallops are hermaphrodites, and so I really, really wanted to maximize how many eggs I got out of the scallops so that I would have enough eggs to do my experiment. But then it ends up being a little bit of a game of risk, because if the scallop switches to spawning sperm, you end up with self-fertilized eggs. That whole set is gone. And so you have to very carefully time it and see, okay, I think this is enough eggs. Okay, I'm going to move it to a new beaker. I hope, and it's just a little tricky, but I will assure you that if I ever did have self-fertilized eggs, they were excluded from the experiment. So I could control the timing of the fertilization to within about 5 to 10 minutes, and that set the timing to 0 hours. Then I added the embryos to the culture container, and this is inoculation. This matters for several of the experiments because that is the time at which they are first exposed to the CO2 conditions. And then finally, I switched the CO2 conditions, and the time for that is the critical factor that varied between the experiments. So to summarize that, I had one set of experiments where the scallops were actually spawned in either ambient or high CO2 conditions, and then the switch and the inoculation time happened at the same time. That's the only case in which the initial conditions were the spawning conditions. Otherwise, they were always spawned in ambient conditions, and then the inoculation time was either 2 hours or 12 hours post-fertilization, with the switches happening at 12 and 24 hours post-fertilization and 3 days post-fertilization. So if we go back and look at the scallop life cycle, we can see that 2 of these timings happened where the switch occurred before the scallops started to calcify. For 2 of the experiments, the switch occurred after the scallops started to calcify. So to check whether or not they actually, at 12 hours, whether or not they had started to calcify, I preserved a set of the embryos at that point and looked at them under cross-polarized light. So cross-polarized light should make any crystallized calcium carbonate birefringe. So it kind of looks like it's glowing. And I did not see that at the 12-hour-old scallops, and so I am working on the assumption that that means they were not calcified at that time point. There is one caveat that I can't really get around, which is that there's some evidence that scallops will form amorphous calcium carbonate before they form aragonite. Amorphous calcium carbonate would not birefringe. So I wouldn't have seen the glowing if they had that shell material at that time. But for the purpose of this, because I didn't see any crystallized calcium carbonate, and because the literature values for this species say that they start to calcify after 12 hours, I'm going to assume at that point they were not calcified. Okay, and then for this experiment, only the 3-day switch experiment, I only had 3 treatments because this was the first one that I did, and so I was maximizing replicates instead of treatments at that point. Okay, so chemistry values. What were the conditions that they were exposed to? So I'm showing you the average pH and aragonite saturation state for the ambient and high CO2 conditions. And what I'd really like you to take home from this is that the ambient condition was supersaturated, and the high CO2 condition was undersaturated. When I compare that to summer values from the 2 locations in Wacoit Bay, you can see that my ambient condition is even more supersaturated than the more exposed site from Wacoit Bay. And the more isolated site from Wacoit Bay was more undersaturated than my high CO2 conditions. I will show you several figures that look like this, so let's just walk through it so you know what you're looking at. These show data from days 1, 3, and 7 of the experiments with the mean plus minus standard deviation. The dashed vertical line, or sorry, the dotted vertical line is the time that the cultures were inoculated. The dashed vertical line is the time at which the switch occurred. When you see treatments with different letters next to them, that means they are statistically different from each other. And then you can see which treatment corresponds to which color. So I'm going to start off by talking about shell size and growth rate. And I would like to point out that, first of all, this work was done several years ago. We know a lot more about how OA affects bivalve larvae than we did at that point, but we already knew that exposure to ocean acidification caused bivalve larvae to be smaller. So my work is not showing, and also there was work that showed changes in shell morphology. The novel aspect of this work is finding the different periods in the larval development that cause those different effects. Oh, so to measure size, at each time point, I preserved larvae and then imaged them and measured the shell length. Okay, so these were the first results, and they were very, very exciting because they show that larvae that were initially exposed to high CO2 are smaller than larvae that were initially exposed to ambient CO2, and that difference persists throughout the first week of development. So by day 1, the 2 treatments that were exposed to high CO2 are already statistically smaller than the ambient treatment. When the switch occurred on day 3, that size difference persisted. So that was very exciting to see. So I followed that up with the experiment that switched the conditions at 1 day old. And again, it follows the similar trend, but now I also know that larvae initially exposed to ambient CO2 and then moved to high CO2 stay similar in size to the larvae that were continuously exposed to ambient CO2. So the results of these 2 experiments indicate that the shell size reflects the CO2 level experienced during the first day of development. So now we'll look at other experiments that help fill out that picture. So from the 2 fertilization experiments, I could see that the CO2 conditions experienced during fertilization did not affect the larval size. So let's focus on the blue and green treatments for a moment. And these are simply 2 iterations of the same experiment. So the blue and green treatments were both fertilized in ambient conditions, and then at 2 hours old, they were put into the culture containers, and for the larvae represented by the green line, their conditions were moved to high CO2. And at 1 day old, they were statistically similar in size to the larvae that had been spawned in high CO2 conditions. So this indicates that it's the exposure after the spawning period that affects the larval size. And because the trends were similar in the two experiments, I am going to focus on the first experiment. Okay, then I did the 12-hour switch experiment, which lets me expand my conclusion that shell size is not reflective of initial CO2 exposure. Rather, it's reflective of the CO2 exposure during the initial calcification. So we can see that from the 12-hour switch experiment. Again, looking at the blue and green lines. The larvae represented by these lines were initially exposed to ambient CO2 from 2 to 12 hours old. Then the larvae represented by the green line were switched to high CO2 conditions. And from that growth from 12 to 24 hours old, they ended up similar in size to the larvae that were raised in the high CO2 conditions since they were 2 hours old. And remember that initial calcification occurs between 12 and 24 hours old. So it's that exposure time that sets the size for the larvae on their first day. And again, the patterns consist throughout the first week of development. And I forgot to mention earlier, there were never any statistical differences in the growth rates from days 1 to 7. So that indicates that the persistent effect of size is a legacy of the difference in size that the difference in growth rate that occurred in the first day. So again, looking at all the shell size results of all of these experiments together, I can conclude that the shell size reflects the CO2 conditions that were experienced during initial calcification. Okay. So we see in the two lower experiments where the switch occurred after calcification had started, we see similar trends where larvae exposed to ambient conditions during initial calcification are larger than those exposed to high CO2 conditions. With the two experiments on top, the switch occurred before initial calcification, and again, it's the exposure to high CO2 during initial calcification that caused them to be smaller. So why is this a critical period? The larvae cannot eat until they're 1 day old, so they are relying on maternally invested energy. We know from other people's work that calcification in undersaturated water is more energetically costly. So while they have finite energy reserves and they're calcifying in high CO2, they end up with smaller shells. After they have mouths, then they can eat and get more energy. And so that is probably why I did not see a difference in growth rate after the first day of growth, since I purposefully fed my cultures and with plenty of food so that food was not a limiting factor. However, in the field, they may not have enough food, and food may be a limiting factor after the first day of growth, and so we may see a different result in the field. So conclusions regarding shell size. Shell size reflects CO2 exposure during initial calcification. Growth rate is not affected from days 1 to 7 at these food replete conditions, and this is likely to be an energetic effect. So moving into shell morphology, these images represent larvae from each time point and each treatment from the 12-hour and 24-hour switch experiments. And they were selected because they're representative of the mean larval size for each time point and treatment. However, we already talked about size, and it's really hard to see the differences based on these images. So what I'd like you to focus on are these dark dots or lines that are located near the hinge. At this point, so I collected these data in 2012, and I had seen one other published study that showed similar images at that point, but they chalked it up to an abnormally formed shell and left it there. So I performed SEM to try to determine what it was, and I could see that it's actually an indentation in the shell. So now I could say, okay, it's causing an abnormal indentation near the hinge. So I went back to my light microscopy images, and I quantified the percent of abnormal shells for each treatment from the 12-hour switch and the 24-hour switch experiment. And you can see that it was the difference between the larvae that were initially exposed to ambient CO2 versus those initially exposed to high CO2 was always highly statistically different at each time point in both experiments. So from this, I could conclude precalcification exposure to high CO2 causes an abnormal indentation near the hinge. Now, why isn't it exposure in the first day? So let's look back at the treatment conditions. And really, we're going to focus on the 12-hour switch experiment here. If it was exposure to high CO2 during initial calcification, we would have expected to see that abnormality in the larvae that were in the ambient to high CO2 switch treatment because they experienced high CO2 conditions during initial calcification. But they did not experience a high percentage of abnormalities. Furthermore, the treatment that was high CO2 to ambient was experiencing ambient CO2 conditions during initial calcification, and they did experience a high percentage of these shell abnormalities. So from these two treatments, the switch treatments, I could conclude that this abnormality is a result of pre-calcification exposure to high CO2. But that's kind of confusing. Why is this happening? That doesn't make sense based on what we know about how larvae are affected by saturation state. And these experiments don't have enough data to provide a mechanism by which this is happening, but we can think about the process involved in shell development. So for bay scallops, when they're about 12 to 14 hours old, they're trochophore larvae, and you start to see the formation of the shell field. Part of the shell field invaginates, and the shell field begins to spread out and has an organic matrix layer that is the periostracum, so the organic layer that covers the calcium carbonate layer of the shell. So during this trochophore stage, the shell field spreads out, it flattens, the organic layer covers the shell field. By the time the larvae are 1 day old, their shell is fully calcified, but it doesn't completely cover the tissue of the larva. And this is called the prodysoconch 1. By the time the larvae are 2 days old, now they're at the D stage, and they have the prodysoconch 2 with this really distinct line between the prodysoconch 1 and the prodysoconch 2. And this is really interesting. But all of this, even the early organic proto-shell formation, is happening after that critical period during which the larvae, when they're exposed to high CO2, end up with that abnormal indentation in their shell. So that exposure period is before any of this happens. And one possibility that I will propose that my work cannot specifically answer is if we think about calcification in echinoderms. So for sea urchin larvae, the mesenchymal cells are ultimately responsible for calcification of the skeletal rods. And they start migration to their final location during the gastrula stage. The gastrula stage would be before the trochophore stage. So if something similar is happening in bivalve larvae, then as the cells that ultimately end up forming the shell field, if they're moving to their location prior to this trochophore stage, then maybe their function ends up being impaired by that early exposure to high CO2, low saturation state. So from the shell morphology, we conclude precalcification exposure causes the shell deformity. So both smaller size and shell deformities can have an indirect effect on survival. So the age at which bivalve larvae metamorphose is determined in part by size, and the smaller larvae may metamorphose at a later age, which increases their risk of predation. And then furthermore, they need to be able to open their shell to allow their velum to come out. So the velum is the organ that allows them to swim vertically and also allows them to gather food. So if an abnormality in the development of the hinge impacts their ability to open their shell and protrude the velum, that might affect their ability to collect food. So I didn't see a decrease in growth that would have been associated with a decrease in eating. However, again, my larvae were fed with plenty of food, and so it's possible that in the field this could cause a reduction in growth rate. So in conclusion, I found two distinct developmental stages that cause very different effects on shells. So exposure to high CO2 during initial calcification causes smaller shells, and exposure to high CO2 before initial calcification causes deformed shells. So this is bad news because it has negative impacts. It could have indirect effects on survival. But there's also good news, because at least in this experiment, with these sets of experiments with bay scallops, exposure to high CO2 after the first day did not negatively impact larval size. So this has implications for hatchery production, where conditions can be much more controlled than scallop larvae growing wild in the field. Okay, moving on to talking about regional efforts in the Northeast, and I will focus on two different efforts. So the Northeast Coastal Acidification Network is a big consortium of groups working together to understand how acidification is impacting the Northeast. What are the geochemical processes affecting changes in carbonate chemistry in the Northeast, and how do those changes affect the organisms in the Northeast? So this group brings together scientists, government agency representatives, fishermen, aquaculturists, and other stakeholders to work together to try to understand this together. In April of 2014, they had a State of the Science workshop where these different stakeholders came together to discuss the changes that we've seen in the Northeast. That workshop ultimately resulted in the publication of a manuscript in the special issue of Oceanography that was devoted to ocean acidification research. And so what I think is unique about this manuscript is that it covers both the changes in the geochemistry, changes in the carbonate chemistry in New England, and also how that impacts the organisms. A lot of times we have the chemistry in one paper and the biology in another paper, and I think it was really great to bring it all together. At the same time that this paper was being written, Maine became the second state to have ocean acidification legislation. So the first state was Washington State. Their Blue Ribbon Panel was much bigger and better funded than Maine's small acidification commission. And like we heard, it sounds like the governor of Washington is very supportive of research. And I would say that it was a big deal for us to get this passed with a pretty conservative political climate and a governor who is not supportive of science in general, and definitely not of climate change science. So this bill passed into law without the governor's signature, but he didn't veto it. So that was a win for us. So this commission, so the commission was a very diverse group of 16 people total, and we only had about four months to complete our work. So it was great that it was happening at the same time as that manuscript with NECAN, because many members were part of both organizations. We were able to learn from both groups. So I just wanted to point out the diverse membership of the commission. So 2 members of the Senate representing both parties, 3 members of the House representing both parties, 3 people who fish commercially, including at least 1 aquaculturist, 2 members of an environmental or community group, 3 scientists who study coastal or ocean acidification, and 3 state agency representatives. So this is the commission makeup that was included in the bill. But when they put together the commission, they were really crafty about it, because one of the, so I represented one of the 3 scientists, but one of the aquaculturists was Mark Green, and he has done very important work looking at saturation state impacts on clams in the sediment. And so, I skipped the most important part. I just assumed you all knew who Mark Green was. But Mark Green is a professor at St. Joseph's College in Maine. So he opened his own oyster farm just a couple of years before this commission was made. So he counted as an aquaculturist, but he also has the science background. So they got an extra scientist. So that was great. And then one of the members of the community group They chose Susie Arnold of the Island Institute, and she also has a PhD in marine science. And so with her, they got a fifth scientist. And so it was really a well-selected group that could provide a very broad range of knowledge. So in January 2015, we released a report. We made 6 recommendations to the state of Maine for ways that they could understand how ocean acidification is impacting Maine's fisheries and come up with ideas for mitigation. And I will admit, we got a lot of our ideas from the Washington Blue Ribbon panel, and we adapted them for the differences in factors affecting Maine. And we really appreciated the work that they had done that we could learn from. However, I think one of the most important aspects of both NECAN and the Maine Ocean Acidification Commission are the new collaborations that they made. And I'm going to give you one personal example. So this is Bill Mook. He is the president of Mook Sea Farm, which is an oyster hatchery and oyster farm in Walpole, Maine. He was a member of the Ocean Acidification Commission, and he has hired me to be the head of his research and development, a brand new research and development program at his oyster hatchery. So this is a personal connection, but I know that there are other new collaborations between industry like Bill and scientists that have come out of both of these organizations. And I think that that was really the goal of these groups, was to create new relationships so that we can work together and learn from each other. Because Bill has already seen impacts of freshwater runoff on his oyster hatchery operations. And so he is very aware that the environment is changing. He's a very savvy businessman. He wants to be one step ahead and know what's happening. And so he's willing to make the investment in a research scientist. So I think that's pretty great. And that is it. In conclusion, we have acidification conditions in coastal areas that are more extreme and variable than in the open ocean. Bay scallop larval development has 2 distinct periods in which exposure to high CO2 has different effects, and regional efforts in the Northeast are being made to reduce local impacts of acidification, because even though it's a global issue, it's one that can be tackled on a local scale. So thank you.